Welcome everyone. In our previous video, we have seen the two types of immunity, that is innate immunity and acquired immunity. In this video, we are going to see the subdivisions under the acquired immunity, that is humoral immune response and cell-mediated immune response. So how these subdivisions came under this acquired immunity? So as I already mentioned in the previous video, the acquired immunity or the adaptive immunity is taken care majorly by two types of cells, that is T cells and B cells. So based on what kind of cell performs the immunity and how it does, we are classifying it to these two types. First, we will see what is cell mediated immune response. So the cell mediated immunity is initiated by the T helper cells. We already know that T cells can be divided into helper T cells, cytotoxic T cells and regulatory T cells. When the T cells take care of the immune response, we call it as cell mediated immune response. This T cell, that is especially this T helper cell, it can help activate the other immune cells and it can target the pathogen to destroy it. In other hand, the cytotoxic T cell, instead of helper T cell, if the cytotoxic T cell is activated, it can eliminate the infected cells from the body by releasing few toxins, thereby promoting the apoptosis or programmed cell death for that infected cell. So as an extra fact, it appears that the T lymphocytes are also capable of recognizing whether the tissue or an organ is from our body or foreign bodies. This is the reason why we cannot transplant and implant the organs into our body even if we find the donor with the same blood group, because our body might reject the transplanted organ. The T cells quickly recognize that the tissue or an organ as a foreign and do not allow it. So it can't become a part of our body. So to avoid it, the transplant receivers have to take the immunosuppressant medication for the rest of their lives, because this immunosuppressant medication will slow down or inhibit the activity of T lymphocyte against this transplanted organ. This response is controlled by the T lymphocytes. So wherever the immune response is mediated by the T helper cells or cytotoxic T cells or regulatory T cells, we call it as the cell mediated immune response. Then what is the second type of acute immunity? So it's humoral immune response. So here the antibodies are produced by B lymphocytes, which is present in the blood cells, and they are transported all over the body. But how this B lymphocytes or the B cells produces these antibodies? So we already heard about antigen presenting cell. So whenever an antigen presenting cell presents the antigen to the T cell can go to any step. For example, the T cell can convert into a T helper cell, or it can convert into a cytotoxic T cell, where it is going to be the cell mediated immune response. But most of the time, this T cell which received the antigen from the antigen presenting cell can go and activate the B cells. So when this B cell gets activated, it can produce these antibodies against the specific antigen. So this is why it is called the humoral immune response, as it consists of an antibody produced by the lymphocytes. It depends upon the action of these antibodies circulating in the body. When an antibody on a B cell binds with an antigen, humoral immunity comes into play. The antigen is internalized by the B cell and presented on the helper T cell. This activates the B cell, as I already mentioned. This activated B cell first will grow and produce a second type of cell called as plasma cell. These plasma cells then release the antibodies in the bloodstream. Sometimes the memory B cells retain the information about the pathogen to prevent any disease caused by that particular pathogen in the near future. This is how the acquired immunity gets the immunological memory character to it. Now I will show you a diagrammatic representation of this acquired immunity and antigen presentation. You can see here, so this red colored thing is a pathogen. So it can be a virus or it can be a bacteria. This pathogen, when enters in our body, the macrophage cells, that is the first line of defense or innate immune cells, these go and attack this pathogen by a process called as phagocytosis. So what is this phagocytosis? So phagocytosis is a process where the pathogen is broken down into components in the phagolysosome. So lysosome is an enzyme which can degrade this pathogen that's present inside this macrophage cells. When this pathogen is engulfed by this macrophage, this pathogen will become a phagosome. This phagosome, when it joins with a lysosome, it forms a complex called as phagolysosome, where the lysosome enzymes are poured over the pathogen and breaks down into smaller components. This macrophage then presents the smaller broken down components of the pathogen to a T lymphocytes, that is T cells. Then this T cell can take multiple paths. For example, when this T cell can activate and become a killer cell or the cytotoxic T cell, which in turn can destroy the infected body cell. Or this T cell can go and activate the B cell or the B lymphocyte, which in turn can become a plasma cell, and in turn this plasma cell can produce these antibodies. So these antibodies are like small missile targets which specifically attacks only the pathogen for which the antigen presentation was performed. So this B cell also can convert into a memory cell which can later induce the secondary immune response upon the renewed contact with the same pathogen in the future. So this is how an acquired immunity performs its function and why it's very important for the survival of a human being.